Okay, so first thing to review is all sponges are multicellular. Okay, they have cells, but they are not organized into tissues and organs like you typically think of when you think of a multicellular organism. Sponges also have specialized cells, just like we do, that perform different functions. Okay, again, key thing is they just do not organize their cells into tissues or organs. Next thing we're going to talk about sponges, what they're made of. Basically, they are a sheet of cells like or similar to our skin over a frame that is like our skeleton. So this skeleton or that structure piece can be made of various substances. You looked at both of these in your activity. So spongin and spicules. Spongin is a soft, flexible protein. It's a network of collagen fibers that make sponges spongy. So when you think of a natural sponge, this is what you're thinking of. Okay, the spicules, they're microscopic structures that are hard and sharp. So these are more glass-like. They can be made out of silica or calcium carbonate, and they also come in a variety of unique shapes. So on the bottom of that page of your notes that says anatomy of a sponge, we're going to go through and add these. First one was spicules. Remember, those are hard and sharp. They're more glass-like. Okay, they're colored orange in this picture that we are looking at. So go ahead and make sure you add that label for spicules. Next piece we're going to talk about, sponges are diploblasts. Okay, remember that means that they have both an outer and an inner layer of cells, but between the two layers, they have a jelly-like substance, which is called mesohyl, which is filled with spongin, spicules, and other cells. So if you head back to your image, the mesophyll is that bluish color, that light blue, right? That's holding the spicules that we labeled along with some other cells within it. Next piece, we've got a couple on this slide. So they're covered in pores. Those pores are called ostia. The ostia allow water to enter the sponge. Okay, so this is kind of allowing that movement of water to start. The large opening at the top of sponges is called osculum. So this is where wastewater exits. So it exits the osculum. And then the large central cavity in a sponge is called the spongocoele. So again, we're going to go back and add those in. We've got the ostia, which are these little pores that are going to allow water to move in. Spongocoele is this large cavity in the middle. It's got that kind of light purple color. And then the osculum is here at the top, and this is what allows that wastewater to exit the sponge. Next, all sponges have a holdfast. Okay, the holdfast is the base that anchors the sponge to a surface. Okay, holdfast would be right here at the bottom, and that should be the last missing label that you had. Okay, moving on to your specialized cells in a sponge. There's a couple different types we're going to talk about. The first is called a choanocyte. These are digestive cells. We also can refer to them as collar cells. They have a ring-like arrangement of short microvilli with a long whip known as the flagellum in the middle. So you can kind of see the structure of one down here. Okay, this is our ring arrangement. We've got microvilli, all these tiny hair-like pieces, and then they have one flagellum in the middle that's coming out. This is going to aid in the movement of water. So in no other animals are we going to focus so greatly on each individual cell. However, because sponges lack true tissues or organs, we are going to focus on their individual cells and the roles they play in their survival of a sponge. All right, talking a little bit more about these specialized cells. So choanocytes, the microvilli trap particles of food. So again, these little hair-like things, that's what's trapping the food particles. And then the choanocyte engulfs it and digests the food with it. The flagellum, so this long tail-like structure that's found in the middle of the microvilli, whips back and forth to create a current of water through the sponge's body. So the key thing about this is it's crucial to their feeding, their breathing, their reproducing, and excreting or getting rid of waste. So memory hook, collar cells create current. Okay, so they help create current, they help with the flow of water so that these particles can move both in and out of a sponge. Similar to what we just did, we are going to go in and label the image. Okay, so first one you're adding are your choanocytes. They are green on our picture. They have, remember, the microvilli and a flagellum on each of them. All right, next specialized cell we're going to talk about are amoebocytes, amoeba-shaped cells that can move through the mesohyle. The choanocytes transfer digested food to the amoebocytes, okay, which carry nutrients to other cells in the body. So memory hook to think about when we're thinking of amoebocytes, this is like DoorDash. Amoebocytes can become other types of sponge cells as well. Okay, so in some species, they do become the sperm cells and egg cells, also known as the oocytes. Back to your labeling picture. Amoebocyte in general, it's got this kind of pinkish color. Okay, it's got that amoeba shape to it, not very cylindrical. And then the oocyte, if it was to be changed into a female egg cell, would be this dark blue. All right, next one we're going to talk about is the pinacocyte. Okay, these are flat. They're like skin cells that are on the outside of the sponge. So you can kind of see a thin layer of them. So if we think about this memory hook, something you can think of is that pin rhymes with skin. Okay, so think pinacocyte, pin rhymes with skin. If you add these labels, again, it's this very thin layer of flat cells on the outside of the sponge. So they kind of have that yellowish color on our slide. All right, we're almost through. Next one we're going to talk about is a porocyte. These are cylindrical cells that make up the ostia or the pores, okay? So they contract and they relax to regulate the flow of water entering a sponge. So again, think of memory hook. Something to refer to a porocyte as is the gatekeeper. It's going to allow or regulate the flow of water entering a cell. If we add our label, porocyte, right, we can see our ostia. The porocyte is going to be those cylindrical-like cells that make up this opening or surround the opening. Cytes make spicules, and the spongocytes make spongin. Adding your label for this one, here is your sclerocyte. It is surrounding that spicule. Next piece we're going to talk about when it comes to our phylum Porifera, or sponges, are body plans in a sponge. So water flow is necessary for a sponge's survival. It's necessary for a sponge to feed, breathe, reproduce, and excrete, which just means to eliminate waste. Every sponge's body is designed for efficient water flow throughout. You can see in the few examples at the bottom of the slide here that both are showing water entering into the sponge in several different places, traveling through that spongocele and then out the osculum at the top. So water flow is something we are going to track on the next couple slides. There are 3 different body plans amongst sponges, and the difference between each plan has to do with their complexity of the canals that are able to move water in and out of the sponge. So if I looked at it, which seems to have the most complex canal system? Hopefully when you looked at it, you looked at the leuconoid. The leuconoid has tons of canals and chambers within it, making it the most complex. Which one seems to have the least complex canal system? If you said asconoid, you are correct. Okay, very few chambers, some canals for them to move through, but very basic when we're talking about that asconoid canal. Okay, before we move on, let's review a couple things from earlier in our notes and from our sponge anatomy activity. So what is this part of the sponge called? Right here. Hopefully you said osculum, right? That is where water exits the sponge. What is the function of the osculum? Hopefully you selected allows water to leave a sponge. On to the next, what are the pore cells called? Ostia, right? Think about what this does. For our next question, which way does water move through the pore cells? Does it go into the sponge out of the sponge or both directions. Into the sponge. The hollow space in the middle of the sponge called? Good, spongocele. Next question, what are the green cells lining the spongocele called? Good, choanocytes. What are two jobs of the choanocytes? Awesome, you could have said create a current, trap food particles, digest food particles. All of those would work for this question. Okay, back to our body plans in a sponge. So if we look at them individually, we've got our asconoid sponges. This is the most basic body plan of a sponge. Choanocytes line that spongocele, and that is where digestion also takes place. So this is the same structure of a sponge that we've been labeling this week when we did that coloring activity. Surprisingly, though, most sponges are not actually this body plan. It is a simple tube shape. These sponges are smaller in size because their thin layers or outer layers break easily, and for one other reason that we will discuss on a further slide. So just remember, wherever the choanocytes are, that's where digestion is going to be. The choanocytes are in different locations for each sponge body plane that we are going to look at. Okay, the biggest thing we'll talk about is that asconoid sponges have the least surface area for nutrient and gas exchange. Okay, so to be a large sponge, you have to have a lot of nutrients and oxygen. These sponges, because they lack that surface area for nutrient and gas exchange, usually are only about 10 centimeters tall or shorter. This is an example of an asconoid sponge. This is a leucoselenia. So you can see how small it is. You can see the simple structure, right? There's not a lot going on with that big osculum at the top letting water leave. The next type we'll talk about, you can hear it multiple ways. So it could be cycanoid or synconoid sponges, but these are a little bit more complex of a body plan. The ostia are going to lead to a network of channels. So you can kind of see here, right? Here's your ostia. Here are your channels that they'll go through. Then you can see them throughout that body plan. And an easy way to think about this one is that it's pleated. So you can kind of see like it's a repeating S shape over and over and over again if you followed along. And don't forget, digestion takes place where the choanocytes are, which happen to be in the canals in this body plan. So you can also notice how much thicker the walls of this sponge are compared to that asconoid sponge we just looked at on the previous slides. And this will allow that sponge to become both larger and stronger. Okay, so thinking about surface area, we've talked about that a couple times. Which of these sponges has a greater surface area for nutrient and gas exchange? And don't forget, surface area includes the parts that contact water, so the outside of the sponge, and more importantly, the parts lining the inside of the sponge, so where the water is going to be flowing through. So not only the outside, but also all of these pathways that the water is going to move through. Good. Hopefully you picked the cycanoid. That one, because of that pleated shape, has a greater surface area. So how will this impact the sponge? Okay, thinking about it, again, more surface area means more contact with water, which means more nutrient and gas absorption. So more nutrients and oxygen will allow these sponges to grow larger and stronger than the asconoid sponges. Okay, so continuing on with our second type of body plan, the synconoid sponges, we just talked about, have more surface area, so they allow more gas exchange and nutrient exchange to occur. The third type we'll talk about is the leuconoid sponges. This is the most common body plan that we see in this phylum, and it's also the most complex. So you can notice all of the canals we talked about, water goes in through the ostia or ostium, moves through the canals, but there's also these chambers that are inside the canals throughout, right? and eventually all still leads to an osculum. This leads to more complex and elaborate channels, which means the digestive chambers, right, are a little bit more complex as well. So if we zoom in a little bit, look inside those chambers. So what do you notice is inside there? What are these little cells called again? Hopefully you said choanocytes. Okay, so leuconoid sponges, the choanocytes are found in the chambers, so not in the canal, right? Not lining the spongocoel, but actually in the chambers. So that is where digestion will take place in this body plan. So which sponge has the greater surface area for nutrient and gas exchange between our leuconoid and our cycanoid? Don't forget, more surface area means more contact with water, both on the outside of the sponge and throughout those channels and chambers. Good. Leuconoid. Awesome. So they have the greatest surface area, right? This means there's more nutrient and gas exchange. These sponges can be huge. So they grow very large. So think large leuconoid. Those go together. An example is a giant barrel sponge. Okay, and here's a picture of a giant barrel sponge. So they are classified as that leuconoid structure. And you can see the walls are very thick, right? If you look at this picture, because leuconoid has the thickest walls. And that is where we will pause in our section of video notes today.